It is about the way to evaluate personalization and evaluation of the best treatment to a cancer patient. So there are a lot of methods which are based on omics approaches, which is genomics, proteomics, algorithms to combine both and basically predict for a patient based on his tumor what would be maybe the best treatment for him. But then again, in most cases, these approaches can result in an unprioritized list of treatments. And eventually, you have to support the physician for the decision what would be the drug to give to the patient, because it's kind of a one-time shot, not like a parallel shots of 5-6 drugs that are recommended by the omics approach. So we believe that the time of the actually wet testing of the tumor tissue of the patient with the different recommendations outside of the patient is appropriate, and this method maybe should be adopted, at least in large medical places, for better treating or better suggesting treatment options to the patients. So what we devised is basically getting a specimen from the patient tumor, maybe during surgery, maybe during biopsy, implant it in mice, immune-deficient mice, getting what it is termed as patient-derived xenograft, but then rather than expanding it and trying to test it in mice, the different drugs, suggested drugs, using just this one PDX and do an ex vivo assay, which can be done about 20, 30, 40 single or combinations of suggested drugs, and within 24 hours after processing this PDX, actually come with a more prioritized recommendation of a treatment to the physician taking care of this specific patient. So this is what I was presenting in this meeting, and I believe that eventually, although the genomics, proteomics, and the other omics approach are developing all the time in order to analyze what would be the best, you still need some kind of wet assay to do it, to get a better evaluation of the personalized treatment. The other thing that I was presenting is the issue of the immunotherapy in this kind of testing approach, meaning when you transfer the tumor of the patient to the mouse in order to do the ex vivo assay after, you are losing the immune system of the patient because it is not transferred to the mouse, only the tumor. So what we have done is to take in parallel from the patient blood, generate T cells of the patient from his blood, and when doing the ex vivo assay, we are also adding the T cells of the patients to the fragments, and then we actually can test combinations of immunotherapy and targeted therapy or single approaches. So we can bring also the information of immunotherapy, which is a very basic pillar in the treatment of cancer today. We can add the information, the personalized tested information also in this approach. We call this approach TEVA, immunotumor ex vivo assay, and we believe that eventually people will work with it in at least large medical centers. The message is that we are willing to teach the protocols, even for people to come to study the method in my lab. We think that this method should be adopted and should be done in different places in the world. This is not something that you can IP or something, so it's a service that we are doing it in Israel with Soroka Medical Center, and we are willing to teach others for that.